next presentation topic will be the neuromuscular research landscape. I would now like to introduce our next speaker, Dr. Heiman Patterson. Dr. Patterson is the professor and director of the Center for Neurodegenerative Diseases at Temple University Lewis Katz School of Medicine. Her career has been dedicated to clinical care and research in ALS or Lou Gehrig's disease. She directs the MDA ALS Care Center of Hope at Temple University and also the MDA Clinic at Good Shepherd Rehabilitation Hospital in Allentown. Under her direction, the Center of Hope has conducted clinical trials testing more than 25 different experimental treatments, while basic research is conducted using a mouse model to study genes that can modify the severity of the disease as well as examine whether novel treatments can extend lifespan. Her clinical research has been directed at extending survival and improving the quality of life. This work has led to studies that examine how to maximize the use of non-invasive ventilation, BiPAP, as well as better address nutritional needs. She has also initiated a program to develop the BCI brain-computer interface for home-based use by people living with ALS in order to allow an increased independence. She has served on the executive board of Northeast ALS Consortium and is a previous co-chair, is a member of the ALS-RG, an international study group, and as president of the ALS Hope Foundation, she is a member of the International Alliance of MND. She has published over 120 papers, abstracts, and chapters on ALS and related motor neuron diseases, along with more than 60 additional papers on other areas of muscle and nerve disease. As an extension of her commitment to ALS, she founded the ALS Hope Foundation, a nonprofit organization dedicated to making a difference to people living with ALS. We are so thrilled to have you here today, Dr. Patterson, and greatly look forward to your presentation. With that, I will pass it over to you.
Thank you so much, but you took up half my time with that introduction.
I'm sorry, you've done so much for our community. It's such a privilege having you here today.
My goodness. Well, it's actually my privilege to be here today. And we're going to talk about the research landscape in neuromuscular disease. And everybody can see my screen now. Is it up? Okay.
Yeah, looks great.
Oh, there we go. So, we're going to start. I always start by talking about what are the diseases that the Muscular Dystrophy Association covers. What do we mean by neuromuscular disease? And then we're going to talk about what's going on. And in talking about what's going on, I want to talk about the tools that we've developed that help us understand disease better, which leads to better treatment. And this is going to include databasing, biomarkers, and also improvements in clinical trial design. And then one of my favorite topics, which is genetics and how an understanding of genetics and how we can manipulate genetic diseases or genetic changes to treat disease. And throughout our discussion of some of the newer treatments and what's out there, I'm going to point out where our understanding of genetics has really led to major advancements and holds promise for the future. And so MDA supports research in diseases that are neuromuscular. Neuromuscular diseases are diseases of what we call the motor unit, which is considered the motor neuron or anterior horn cell as it comes out of the spinal cord, and the muscle fibers that it innervates, and also that communication point between the anterior horn cell and muscle, which we call the neuromuscular junction. And so in terms of diseases that can affect that anterior horn cell, that lower motor neuron, and the sensory component of the neuromuscular system, we have spinal muscle atrophy, which we're going to have some exciting discussion about the treatments there, Charcot-Marie-Tooth, ALS, and then Friedreich's ataxia can affect that. Then in terms of the neuromuscular junction, myasthenia and Eaton Lambert, these are autoimmune diseases where our own bodies produce something called antibodies, which attack that junction in different regions. And then muscle. And in muscle, we have inherited diseases, which are all the dystrophies. We have inflammatory disorders like polymyositis and dermatomyositis. And then we have metabolic problems with the way muscle makes energy. Muscle makes energy by burning sugars and fats in a furnace we call the mitochondria. So we're going to talk a little bit about some of the advancements in each of these areas. And we're going to start with one of the things is that we're establishing tools that enable us to advance understanding mechanism and understanding treatment effects. And one of the tools that's being developed has actually been funded by MDA, which is MOVR, which is a database, a large national database from MDA clinics. And it's really designed to enhance management and care and understand better natural history of disease and effects of different medications on disease progression. It also will aid, in addition to providing natural history, which will help in design of trials, it will also aid in identification of people who might qualify for trials in some of the rarer neuromuscular diseases. Where do we find them? Where do we find people with what's called Pompe's disease, which is a pretty rare disorder? And right now, it includes ALS, Duchenne, Becker's, FSH, limb-girdle dystrophies, Pompe's, and SMA. But the plan, of course, is to enlarge and continue to add additional diseases that we can track and develop data sets for. Very important. The second important area that enables us to develop an understanding of disease and treatment modalities is the development of biomarkers. A biomarker is a measure of a state, a biological state, or a disease process. It can allow earlier diagnosis. If you're measuring sugar in a diabetic, an elevated sugar tells you that somebody is diabetic and can track the progression and treatment effects. And that's a biomarker of diabetes. So some biomarkers can allow diagnosis. They can also help us look at progression of disease. There's something called neurofilament in ALS, which as nerve cells die, there's an increase in neurofilament levels. And we're working on that as a potential biomarker that tells us about progression. They can help us understand mechanisms. And if we understand a mechanism, that can help us to stratify people for treatment strategies, because you want to direct the treatment at a mechanism, so you want to know what the mechanism is behind a person's disease. And it can help tell us if the drug got to where it should go and had the effect it should. So for instance, in a diabetic, if I give them insulin, what happened to the sugar? The sugar should go down. That tells me that the insulin got where it was supposed to go and dropped the sugar. And what kinds of things could be a biomarker? Well, you can have imaging. MRIs can tell you that there's been a stroke. PET scans are a way that we can look for amyloid in Alzheimer's, for instance. Or there's markers for inflammation in diseases with neuroinflammation. ALS happens to be one of those. They can be biofluid, spinal fluid, serum, and plasma, and DNA. And all of these can be looked at for markers of disease activity or diagnosis. It can be a biopsy of a tissue, a muscle biopsy that tells you there's inflammation, for instance. And finally, certain clinical tests can look at progression. For instance, maybe a functional assay to tell if a disease is changing function over time. That would be a biomarker of disease progression. So all of these are important because we need to be able to diagnose early, follow progression of an illness, and know that we did what we were supposed to with a drug. So they're all critical pieces, and understand the mechanism so that we can stratify properly. These are all critical pieces for being successful in our trials and finding drugs that truly help. And what is the goal and challenge of clinical trials? Well, it's to find treatments that stop or reverse a disease that are safe and truly effective, right? The challenges are what I've been talking about, targeting the treatment and understanding mechanism. That's where biomarkers help. We need better biomarkers of both efficacy, target engagement, as well as diagnostic and stratifying. and helping us to choose the appropriate test population, and we need to choose the right endpoints. What's the right question to ask? Is it survival? Is it function? What do we want as our primary endpoint? If we treat someone with a disease, what is the outcome we're looking for most? And there are several phases to clinical trials. We start with phase 1, and that basically, and this is in human, this is after years of preclinical work in animal models and tissue culture, and we finally get a drug that we're going to take to people. The first phase is phase 1. It can be in healthy individuals, and we basically want to make sure that we don't kill anyone. That's the most straightforward way to say it. We want to test for safety and look at some dosage and what a particular dosage, what the level is in the blood or the spinal fluid of a particular drug. Phase 2, if the drug looks safe enough, we take it to phase 2, where we actually put it into more people so we can get more information on side effects. And that's when we start to look at whether or not there's a hint of efficacy, if we should take it forward to a major phase 3 trial. And so typically phase 2 are not powered. There aren't enough patients in the trial to look for efficacy, whether or not the drug works, but just enough to figure out if we should take it forward, because these are expensive trials. Phase 3 is where we do a full-on trial. We have, typically there will be treatment and placebo groups, and it's powered so that we can detect an effect if there is in fact an effect. Now, all of these phases, 1, 2, 3, all of that can take 10, 12 years. A phase 3 trial can typically run 2, 3, 4 years. And I'm going to bring that up when I talk about changes in design of trials, which have really been piloted by cancer treatments, but now we're moving into neuromuscular disease, ways to speed this up and actually minimize your chance of being in the sugar pill or placebo group. And once you get past phase 3, if you're successful, you go for FDA approval. And after FDA approval, there's real world evidence where we really look at what's the effect in the general population as we treat people outside of the clinical trial parameters. And is it still effective, and is it still safe? Is there anything we didn't notice in the trial that comes out? Now, I promised that we would talk a little bit about one of the changes that's occurring in clinical trials. It's going to speed things up. As we have more drugs to try, if I do a conventional trial, it's one drug versus placebo, and it takes, like I said, 3 or 4 years for a phase 3. The platform design enables us to test many drugs at once. The platform trial in ALS has tested 4 drugs together at the same time in parallel. All of the drugs companies whose products we're testing have to agree to the same protocol. They have to agree that they want to do it all the same way. And the placebo group is a shared placebo. What that means is if everybody's sharing the placebo group, there's less chance, if you're a participant, of receiving sugar pill. In addition, I'm testing 4 drugs here in placebo, and this will take 2 to 3 years, and I'll get 4 drugs tested. And as we test, as one drug is successful or unsuccessful, we can replace it with another drug. It's adaptive, meaning we can drop ineffective treatments, add new ones. It's more efficient. And as I said, it shares the placebo group, making it, a lot more efficient and actually patient-centric, thinking about the patient. Who wants to be in a sugar pill group, right? So it's very patient-centric. And by the way, many of our endpoints, and I didn't mention this before, when we do the phase 3, at least in ALS, we have really incorporated the opinions and feelings of people living with ALS about what they want from a drug and how they see design, and trying to be very forward thinking about stakeholders, the people who this really matters to, which are people living with the disease, what their feelings are. This is important that we engage everybody in the design and implementation. Now I'm going to move into a 4th area kind of where I think it's made a huge difference in the progress, and that's genetics. And I'm going to start with our little genetics primer. We have 22 chromosomes, 22 pairs of chromosomes. Each pair has one chromosome from mom and one from dad. And then we have our sex chromosomes, which if you have an X and an X, you're a female, and an X and a Y, you're a male, and you get the Y from dad always. So, but you get half of your chromosomes are from mom and half from dad. And each chromosome contains the blueprints for proteins, which are called genes. And that blueprint is in the form of a language, the language of DNA, which is a sequence of, you can think of them as letters, but they're nucleotides. And there are 4 different nucleotides in DNA: guanine, cytosine, adenosine, and thymine. And this sequence is transcribed into what we call RNA. And the RNA then goes out into the cell machinery and pieces together the building blocks of protein, or gives the information to the machinery that makes proteins what the sequence of these building blocks, these amino acids, should be, and that then builds the protein. And 3 of these nucleic acids determine one amino acid. So that sequence is the blueprint for how to put together a protein. But things can go wrong, and these are called mutations. And so if you look at this sentence, these are 3-letter words, and to denote the 3 nucleotides here's normal. DNA has all you can ask for, but sometimes you can have a change in one of those amino acids that doesn't make a difference. One of those nucleotides rather, that doesn't make a difference because the same protein building block will be put in and it sounds the same. So this looks the same. Or you can have a nucleotide replaced, or a mutation that changes the sense of the sentence. DNA has all Lou can ask for. Well, that changes the whole sentiment, the whole sense. Or you might put in something that just stops, and creates a stop codon, which stops the translation to a protein, and we call that a nonsense. Or you can add a nucleotide, which really screws things up because everything downstream is shifted, and we call that a frame shift. And that makes the protein downstream all abnormal, right? All the wrong sequences of 3 will be decoded and translated into the wrong protein, the wrong protein building blocks. And so these are the kinds of things. And so we have sort of developed strategies for many of these abnormalities that we're going to talk about as we go over some of the genetic changes. For instance, when there's a stop codon, there are strategies that skip over it and reestablish the reading frame. We have ways to block if a protein is made because of this missense into a protein that's toxic. We can block the production of that protein. We can replace a protein if it's not made at all. These are all strategies based on the genetic mechanisms and that are a reality now, that are in trial or even been approved for treatment. And how do we inherit a disease? Well, from our parents. And there are 2 major forms of inheritance I wanted to mention: dominant and recessive. In dominant, you only need 1 bad gene. You can see this little dark blue gene, 1 bad gene that you inherit, and you will have the disease. So if your parent has it, you have a 50-50 chance of getting that bad gene and then having the disease, and a 50-50 chance of being unaffected. In the case of recessive, your parents actually carry a bad gene but don't have the disease because you need 2 damaged genes to have it. And in this case, you need to inherit the bad gene from both parents, that damaged gene from both parents. And that happens only 25% of the time. But half the time you will inherit one damaged and 1 undamaged gene, and you will be a carrier. So if you were to have children with someone who also carried that disease, you would then have the risk of passing it on. Otherwise, you would not. And you have a 25% chance of being totally unaffected. So that's recessive, 2 copies of a damaged gene, whereas dominant, you only need 1 copy. So let's talk a little bit about genetic manipulations that are being used that are either in trial or actually have been approved. So we can deliver through genetic means recombinant neurotrophic factor, and that's being tested in Charcot-Marie-Tooth, where this is a factor that is nutritional for nerves and helps them do better, helps them survive better. We can block a toxic gene. We're blocking the SOD1 gene using a strategy where we block RNA from becoming protein, something called antisense. So we block that RNA so it isn't translated into protein. In Duchenne, there are several pharmacologic agents now approved that enable a skipping over an area of deleted or damaged DNA so that the DNA reestablishes the reading frame and a whole protein is made rather than being degraded. And in addition, there's attempts to deliver both a mini dystrophin and an alternative protein that can substitute for the dystrophin protein that's damaged in Duchenne muscular dystrophy. There are also, in Europe, it's been approved, a drug that actually reads through a stop codon, pushes past that stop codon to reestablish reading. Myasthenic dystrophy, there's some strategies developing that block some toxic regions of that RNA, remember DNA to RNA. Limb-girdle dystrophy, there's gene replacement therapy that's being tested. In oculopharyngeal dystrophy, there's also some testing of RNA interference to block the production of abnormal protein. And SMA has probably been our biggest win, if you will, where we've changed RNA splicing to create replenishment of a protein that was missing. And really, it's made a world of difference. It just turned the world of SMA upside down, and also the ability to deliver a normal gene to replace the damaged gene. And these are approved. They're being used in treatment now. So let's talk a little bit about some of the diseases. We'll start with Charcot-Marie-Tooth, ALS, and spinal muscular atrophy. In Charcot-Marie-Tooth, there are two types. Type 1 is damage. Think of the nerve as a wire. We have a wire and an insulation. In type 1 Charcot-Marie-Tooth, there's damage to the myelin. This tends to be a dominant. In type 2, there's damage to the wire. And this can be either dominant or that recessive style of inheritance. It causes progressive weakness and sensory loss starting in the legs. We've now identified more than 20 different possible genetic causes. And there are several clinical trials going on, some of which are using the genetic information that I just mentioned. In one case, we're delivering a neurotrophic factor to try to salvage nerves, to give them chicken soup for nerve cells that are dying. In addition, there are several other trials going on, and in preclinical trials, there's some gene replacement studies going on, hopefully that will come to people and be used in full clinical trials in humans. ALS is another disease that's been very exciting. There's damage to both the upper and lower motor neuron. The motor neuron that carries messages from the brain down through the brain stem into the spinal cord is the upper motor neuron, and then the lower motor neuron is that anterior horn cell of the motor unit we talked about. And there's damage to both of these motor system cells, and that causes atrophy, thinning of the muscle, weakness, twitching, stiffness, and reflex abnormalities where the reflexes are too good, and people can present with arm or leg weakness or trouble swallowing. It is progressive. 10% of folks have a family history of ALS, and in that, those people with a family history, we've identified more than 40 genes that have been associated with ALS. The two of the most important ones are C9 open reading frame 72, which has an extra portion of DNA. And one of the strategies is to block that extra area of DNA because it makes an abnormal protein called the dinucleotide repeat that gloms up the cell. SOD1 was the first gene ever identified. And by the way, C9 accounts for 40% of that 10% familial. But of interest and import is that it's also found in people without a family history, up to anywhere between 4 and 10%, depending on the population. And SOD1 makes up 20% of familial, but it makes up 1 to 2% of sporadic cases. People without a family history, you can also find it. Another gene that's been important is TDP43 that isn't mentioned here, but TDP43 is a rarely mutated gene in familial ALS, less than 1%, but aggregated TDP protein is found in virtually all ALS folks, whether they be familial or they be sporadic out of the blue. And the importance of these 40 genes that have been identified is that they are teaching us about motor neuron biology gene by gene. And so each of these genes, when you look at the pathway or the mechanism that they damage, you can see virtually everywhere in the nerve cells. And the big ones that have been identified that now are targets for treatment include the way DNA and RNA is processed, protein aggregation, neuroinflammation, oxidative stress, and transport of things up and down the neuron. And I just show this to show you how every area of the nerve cell can be affected by these 40 genes. And like I said, we're learning motor neuron biology gene by gene. And this just shows you, based on these mechanisms, all of the drugs that are in trial. And we have 2 drugs that have been approved in red, Radicava and Riluzole, that slow the disease down, and Nudexta, which helps with some of the symptom management. But I've shown in green the things that are now being tried in the platform trial, everything at once. So that's one of those adaptive trials. AMX 035 is going into phase 3, and it had a successful trial that showed efficacy and actually is up for approval in Canada and may get approved without a phase 3 trial, but the phase 2 was so encouraging. And then, of course, as I mentioned, there's the gene modulation, both aimed at the C9, at another gene mutation called FUS, and SOD. And the SOD just finished a phase 3, and with antisense blocking the production of SOD1 because it's a toxic gain of function. That means when it's mutated, this gene produces a protein that's actually damaging to the cell. So blocking its production might be useful. And there's a lot of hope. Oh, there we go. Spinal muscle atrophy, extremely important to talk about. This has 3 variants: infantile, juvenile, and adult presentations. And we'll talk about why that is in a moment. It's a recessive disease. And as a result of the mutation, there is no SMN1 protein made. It causes weakness and breathing difficulties. And in the newborn period, it's really quite progressive, and most infants die before the age of 2 because of this disease. Our treatment previously was, make sure of the diagnosis, take care of breathing and nutrition, keep people as functional as possible with physical therapy. But now we've entered the time frame of genetic modulation, and we have gene replacement, Zolgensma, which actually replaces the SMN1 gene. And then we have strategies aimed at leveraging the SMN2. And SMN2 is our genome contains SMN1 and multiple copies of SMN2. SMN2 looks exactly like SMN1, except for one problem. This exon number 7, labeled T, gets spliced out, cut out when the protein's made. And therefore, the protein that's made is only partially functional, is rapidly degraded, and only 10% becomes good protein and is exactly the same as SMN1. In SMA disease, there's no SMN1, and you're relying on whatever protein is made by SMN2 to salvage you. And the difference between those different types of SMA, the adult versus the juvenile versus the severe infantile, is how many copies of SMN2 you have. The more copies you have, the more of this partially translated protein you make. So this if you have 4 copies, you'll have about 40 or 50% of what you should have of SMN1, and therefore your disease is milder. Now, the strategy that's been used, and the first drug that was approved was nusinersen or Spinraza, where it was an antisense that kept that exon in. That exon that's cut out is kept in, and you get a full-length SMN2 becomes a full-length SMN protein, so that you actually get the more of that normal protein made. Subsequently, we have an oral ASO, which is Evrysdi, and then I mentioned Zolgensma, which is actually replacing that SMN1 gene with a viral vector. Very exciting, has changed the landscape of SMA. The kids are thriving, gaining milestones, especially juvenile, but even infantile children are living and stabilizing. And this is very exciting to those of us doing neuromuscular disease. Next, we'll talk about the neuromuscular junction. And the 2 big diseases are myasthenia and Eaton Lambert. These are autoimmune diseases. In the case of myasthenia, your body makes antibodies which damage these receptors. You know, we have, when electrical activity comes down the nerve, that's called the depolarization, these packets of transmitter, a chemical, are released and float across the junction and hit on these acetylcholine receptors. And the chemical is called acetylcholine. And that sets up the muscle to contract. That changes the muscle membrane potential and causes it to contract. In myasthenia, the antibody is directed to this receptor so that when the acetylcholine is released, it can't really communicate well. In the case of Eaton Lambert, the antibody is to this calcium channel, and what that does is reduce the chance that you'll release a packet of acetylcholine. So it decreases the release of acetylcholine. To start with myasthenia gravis, it generally can affect the face and neck as well as the eye muscles. It's more proximal. It can include swallowing and speech. Respiratory muscles can be affected, and there may be a genetic predisposition. And medication, the therapies include medications that block the breakdown of acetylcholine. This is mestinon, and therefore it hangs around longer so that it can find receptors that aren't damaged by this antibody attack. But the real treatment is aimed at that autoimmune disease. And so we've done treatments like pheresis and IVIG, which take those antibodies out of the blood and take them out of circulation. But in addition, we now have Solaris, which actually blocks complement. Let's see if we have it. There it is. It blocks complement, which blocks the sort of autoimmune attack on those receptors. And in addition, there are trials of several other drugs like Solaris, which also affect this cascade, the C5, complement 5, as well as what we call the fetal receptor, which alters autoimmune. And one drug is already at the FDA under review. So pretty soon we'll have a whole arsenal of medications that we can try when steroids and pheresis and IVIG aren't effectively controlling disease. With Eaton Lambert, as I mentioned, it's these voltage-gated potassium channels and calcium channels that have the antibodies. Here you go, right there. And again, this is antibody-mediated. And the drug that has been approved recently is Firdapse, which blocks the voltage-gated potassium channels and sort of evens out the problem so that these packets of acetylcholine can be released better. So a new drug. In addition, like myasthenia, given this is autoimmune, people have used the pheresis, the IVIG, and prednisone to treat LEMS as well. And now moving on to muscle diseases. We're going to start with the dystrophies. And I'm going to be pretty fast because I don't want to run out of time. But Duchenne is an X-linked disorder, and I know many of you are familiar with this. It gives elevated CPK. It causes progressive weakness with sometimes enlarged calves and contractures. And of course, we have standard treatment with physical therapy. We want cardiology involved, and we want respiratory care. And we want to optimize independence. And we have, of course, steroid deflazacort and prednisone, which has been shown to keep boys walking longer. Idebenone has been shown to maybe improve breathing function. And atelurin, I mentioned before, reads through that stop codon, and that's been approved in the European Union and United Kingdom, but not in the US. But what we do have is exon skipping. So you can think of it as a Scotch tape. Normally, there's a deletion or a stop codon that would stop things right here. And in this case here, we have exon 51, say, but this allows a read through over that damaged area. And in 2016, we had our first exon skipping drug, eteplirsen, but now we have 3 others that have been approved. And of interest, these were approved based on what we call biomarker, because they showed a significant increase in the amount of dystrophin made. Normally in Duchenne, there's no dystrophin in the muscle because of the damage. But if you can increase dystrophin, the idea is that that would indicate that you would improve muscle function. And there have been several of these exon skipping agents that did not necessarily show a clinical effect, but showed increase of dystrophin and were approved based on what we would call a biomarker and the assumption that that would translate to a clinical effect long term. In addition, we have some trials where we're doing surrogate gene therapy and where we're trying to insert genes that could help the dystrophin that's there function better, and also a mini dystrophin construct. And another strategy is to upregulate a protein called utrophin, which is very similar to dystrophin and might substitute for dystrophin. Limb-girdle dystrophies, this is a huge group of diseases. There's recessive and dominant variants, and there's probably over 30 or 35 different genetic mutations now identified and associated with it. They're variable in terms of their clinical picture, but generally their progressive proximal weakness onset can vary. They may have heart and lung involvement. Treatment has been supportive, but our research is based on, of course, MOVR and the natural history, but clinical trials with gene therapy. And so we're doing gene therapy for particular mutations, which is really personalized medicine. And as we get more successful with these, we'll be able to design gene therapy for virtually any of the genetic mutations. Myotonic dystrophy, another very important disease. It's the most common of the inherited adult dystrophies. It's dominant. It causes weakness in the hands and feet, which is distal, and facial weakness. It can involve every body system. And the reason it's called myotonic dystrophy is myotonia, which is the inability to relax a contracted muscle. The phenotype can vary, and it's due to extra DNA. So in this myotrophin protein kinase gene, there's extra DNA that doesn't belong there called a trinucleotide repeat, and that leads to extra loops of RNA. And these extra loops of RNA impact the function of other proteins by binding them. So in this case, muscle-blind protein, another important protein for muscle, gets glommed onto there, can't go do its job, and that causes the problem. And here again, you see another depiction of this muscle blind getting stuck to the RNA where there's extra RNA. And here's the repeat fragment. The strategies that are very exciting include an antisense strategy to block the binding of this muscle blind protein to, here you go, to the loop of RNA. And in addition, there's a monoclonal antibody that's conjugated with a small RNA that interferes with the production of this area of RNA. And that's actually in clinical trial. AOC 1001 is in clinical trial now, and we're all crossing our fingers hoping that it works. So we are making progress. There's a second type of myotonic dystrophy, type 2, that's similar to type 1, but it's a little milder. And it's due to also extra DNA, but in a different protein. And we don't have any genetic treatments there yet. And it's mainly the supportive care that we give to all folks. And you can treat the myotonia with mixilotine. FSH comes in 2 varieties. It's the most common is type 1. It's a dominant. It's called Facioscapulohumeral because it involves the face and the shoulder blades also can have lower leg weakness and then proximal weakness. You can have heart and respiratory muscle involvement. The FSH1 is due to a decrease in the number of a repetitive element in this chromosome area, D4Z4. So this area of the chromosome has a reduced number of these repeats, which results in a protein that's nearby called DUX4 being decreased, or increased rather. We don't know the function of DUX4, but we do think that this causes a toxic gain of function. And so treatment strategies are directed at reducing DUX4. In the case of the second type of FSH, the problem is in chromosome 18. And while the number of these repeats is normal, DUX4 is still increased. And again, we're hoping the same strategies of blocking DUX4 will work. And so Fulcrum Therapeutics has developed a small molecule to block that DUX4. And there are several other trials that are going on in FSH. So again, exciting, we're making real progress. Similarly, oculopharyngeal is a rarer muscle dystrophy, has eye muscle involvement and proximal muscle, and it's also caused by extra DNA in a gene, the PABPN1 gene, that results in a non-functional protein that then ends up clumping or aggregating. And again, there is a DNA silencing strategy being aimed at oculopharyngeal. Big progress. If you understand the genetic change and you understand the mechanism of that, what that does, you can design strategies to treat it. And finally, Pompe's, which is an infantile disease that causes loss of acid maltase, we have repletion of the enzyme that's missing. And again, we've made this has been a major stride forward in the treatment of this disease, which was rapidly progressive. Now, I'm going to end on the inflammatory myopathies, polymyositis and dermatomyositis. This is an adult disease. It's inflammatory and autoimmune. It's really progressive proximal weakness. In the case of dermatomyositis, it includes a rash. They have different immune mechanisms, but they both cause inflammation in the muscle. We have a lot of treatment strategies. We've used prednisone and immunosuppressive agents along with physical therapy and attention to cardiac and pulmonary complications. But there's just a host of trials of new anti-inflammatory and immune modulating agents that are now being looked at. And so the pipeline is large. And similarly, inclusion body myositis, most frequent muscle disease over the age of 50, especially in men. The onset it's very classical. It causes forearm weakness and an inability to make a fist with proximal quadricep weakness. You can't kick out your leg or lift it to march. And again, we're making some progress here as well with many clinical trials going on. And hopefully we will get to the point where one of these pans out in phase 3. The one phase 3 is a beta sept right now. And so we've had unprecedented progress towards treatments. And this is really the underpinning of this progress has been understanding the genetics and developing genetic approaches for genetic diseases, as well as understanding mechanisms and targeting treatments to the mechanisms in both genetic and autoimmune diseases. And we're developing tools that track disease and responses with biomarkers and which can speed up trials and help identify the proper people to try drugs on, new assessments, and our natural history database. And with that, I'll stop and say we've only just begun. Thank you for your attention.
Thank you so much, Dr. Patterson. We really have only begun, and hopefully we do have some questions that come through. I don't see any yet, but we do have about 3 to 4 minutes here. If anyone has questions, please do put them in the chat or in the Q&A. Okay. Looks like one came through. Will there ever be any testing with LGMD? I have 1B.
Absolutely. I showed, I think that there are several gene replacement trials going on. And if in fact we're successful with one, those strategies can be translated to all of them. So gene replacement is going to be, we're doing that in several of the limb-girdle dystrophies. And once we're successful, we can do it with any of the limb-girdle dystrophies. One of the caveats, though, is that we have to understand the mechanism of the change. So we have to be very careful when we do gene replacement. Sometimes we may have to knock down the abnormal gene and then replace. We may have to cut it out with something. I didn't have time to go into what it's called CRISPR. You can snip out bad genes and then replace them with good genes. And that's being done in the animal models. And there's actually been some attempts at CRISPR treatment in other diseases, not MDA diseases, but other diseases. And again, the lessons learned will be translatable to neuromuscular disease. And, in ALS, which of course that's my passion, in ALS, we're now doing personalized treatment based on your genetic mutation for FUS mutations. That's going to happen with the limb-girdle dystrophies. And, and I didn't get a chance to talk about limb-girdle dystrophies in depth, but they're great because all those different mutations have taught us so much about muscle structure, because all of the proteins that are mutated in limb-girdle dystrophy relate to how that muscle holds together, the muscle cell membrane and the structure that keeps muscle from, just imagine when you contract and relax a muscle, that cell surface has to be really strong not to break apart. And the genes involved in limb-girdle dystrophy all hold that muscle together. And they also stabilize the muscle during contraction with dystrophin. They're hooked onto dystrophin. So, they're not so far from Duchenne dystrophy. So I could go on and on, obviously. There might be another question.
Yes, there are lots of questions suddenly, as I welcomed questions. I do unfortunately probably only have time for one more question. So we will address, I have DM2, and you mentioned that no real research is being done for that. Do you anticipate that may change?
Yes, because DM2 is also, a trinucleotide repeat. And as we understand better trinucleotide repeat diseases, I suspect we're going to have strategies that target those trinucleotide repeats genetically. So while it's not being looked at now, it is, but while we don't have, it is in the sense that we're creating natural history, and that's really important, and diagnosing it and getting groups of people that could be used for a clinical trial. We don't have a clinical trial yet, but I don't think it's going to be too long before somebody starts looking at the trinucleotide repeats in DM2.
All right. Well, I think that kind of wraps up our time for Q&A today, but thank you so much for being here. Dr. Patterson, we will definitely welcome you back in the future. We have many thank yous going on in the chat, so I know that the participants also appreciated your presentation. So thank you so much.
If people have questions, they can send them to you and you can pass them on to me, and I'll answer them and send back.
Wonderful. That's so appreciated and very generous of you. So thank you very much. I will do so.
My pleasure. Thank you.